Assessing PFAS exposure risks in threatened and endangered species, or T&E species, is currently a critical priority for implementing effective management of AFFF-impacted DoD sites in the United States. The objective of the present CERTUP project was to develop a framework for conducting robust, scientifically defensible risk assessments of PFAS in T&E species at impacted DoD installations. The present study provides a comprehensive literature review of physical-chemical properties, bioaccumulation metrics, and environmental concentrations. The proposed approach generally follows conventional methods employed for ecological risk assessment of chemical contaminants, including exposure characterization, effects characterization, and risk estimation. Specifically, the proposed approach involves utilizing a combination of field-based measurements and bioaccumulation modeling to evaluate PFAS exposure in T&E species. Toxicity reference values, or TRVs, are derived using the available toxicity data along with species sensitivity distributions, or SSDs, and resulting fifth percentile of the hazardous concentrations, or HC5s. When there is a lack of toxicity data, TRVs are derived using the lowest single-point toxicity value for a given organism class. In the proposed framework, we also demonstrate the utility of using chemical activity for PFAS exposure and effects characterization. Chemical activity in a given medium is determined as the ratio of the chemical concentration in moles per meter cubed and the corresponding solubility S in moles per meter cubed of the chemical in that medium. The chemical activity-based approach provides a thermodynamically sound modeling framework for describing the behavior of PFAS in environmental and biological systems. We developed a mechanistic PFAS food web bioaccumulation model to predict internal exposure levels, concentrations, and activities, as well as external exposure and dose estimates in various aquatic and terrestrial organisms. The model was parameterized and applied to simulate PFAS bioaccumulation at several DoD sites that have existing PFAS monitoring data. The following are some of the key findings, uncertainties, and knowledge gaps, as well as recommendations for future research efforts. Based on the available toxicity data for aquatic biota, a concentration-based TRV for PFAS exposure in aquatic biota was determined to be 1.1 microgram per liter in surface water. This corresponds to a chemical activity equal to 1.6 times 10 to the negative 6. The available PFAS monitoring data for several DoD sites indicates these TRVs are in some cases exceeded. Based on the available toxicity data for birds and mammals, an external dietary dose-based TRV for PFAS was determined to be 20.5 microgram per kilogram body weight per day. Also, based on observed PFAS serum concentration related to reproductive impacts in birds, a serum concentration of 0.25 microgram per milliliter was derived as an internal concentration-based TRV for birds and mammals. Available monitoring data and model estimates indicate that PFAS exposure in wildlife at AFFF-impacted DoD sites may in some cases exceed the derived TRVs. The mechanistic PFAS food web bioaccumulation model was shown to predict internal PFAS exposure levels in biota at DoD sites reasonably well, with model prediction values generally within a factor of 3 of the observed field data. Model simulations of perfluoroalkyl acid bioaccumulation in aquatic and terrestrial food webs demonstrate that these compounds are expected to preferentially biomagnify in food webs containing air-breathing wildlife such as birds, mammals, and terrestrial reptiles. Conversely, perfluoroalkyl acid concentrations in aquatic water-respiring organisms are expected to reach a chemical equilibrium with concentrations in ambient surface water. In other words, PFAS activity in aquatic biota is equivalent to that in the ambient surface water. PFAS concentrations in soils was found to be very important for exposure risks in numerous T&E species within terrestrial food webs, including terrestrial reptiles and birds such as the eastern Massasauga rattlesnake and Kirtland's warblers. It is important to note that risk estimates for T&E species in the present study are based on scenarios that assume exposure occurs via concentrations at the studied DoD sites. The extent of interaction of T&E species at specific DoD sites is a major knowledge gap in the present assessment. Future research efforts should include investigations to better understand the frequency and duration of various T&E species at AFFF-impacted DoD sites. In particular, studies to determine PFAS concentrations in prey and relative prey consumption rates at these sites would be useful. Also, there is a current need to better understand PFAS sediment and soil residue concentrations at DoD sites and corresponding exposure risks in upper trophic terrestrial wildlife. Lastly, PFAS solubility estimates are essential for effective development of mechanistic bioaccumulation models and application of the chemical activity-based risk assessment approach. Currently, there is a need for further laboratory-based measurements of PFAS solubilities and related distribution coefficients for different biological media, including proteins, phospholipids, and neutralipids.